Well, have a good day. Today, Thursday, December 15, 2022, we want to welcome you very warmly to this webinar on Chlorpyrifos, harm to environmental health, evidence for its banning in Mexico and worldwide. My name is Fernando Bejarano. I'm coordinator of the IPEN Office for Latin America and director of Rapam in Mexico. On this occasion, we have a magnificent panel of experts. This is the second webinar that we are organizing. It's part of a project supported by IPEN for Latin America in order to carry out diagnosis of the situation of chlorpyrifos in different countries. The webinar, the last one we had the participation of Chile, Uruguay, and this time we will focus on the situation in Mexico. We have, therefore, 2 hours for this meeting. First of all, we have 2 experts who will speak in English. We've given the instructions for you to choose the interpretation mode if you need it. And we will therefore start. We have a couple of other participations. First of all, Sara, Brosché, and Megan Horton, and then we'll open up for questions and answers. For the questions, you can make your questions, you can ask your questions in the QA option or in the chat itself. So let's start. We will start with the first presentation. We have with us Sara Brosché. I'm going to read some information from her CV. She's scientific advisor of IPEN. She gives technical, scientific, and political support and strategic support within the framework of the Stockholm Convention, the Basel Convention, and SCASM. Sara started to work with IPEN in 2012 after having done her MA in chemistry and a PhD in toxicology from the university. She's been involved in the review committee of COPS since 2016. And she's also been involved in the project to eliminate lead in paint. It's our pleasure, Sara, for you to be with us today from Sweden. And please, can you activate your camera and share your presentation?
Thank you very much, Fernando, and
Muchas
good morning,
gracias, Fernando.
everyone.
Buenos días, todo el mundo.
I can't seem to be able to activate my camera, but I will share my screen.
Pero voy a compartir mi pantalla.
Okay, I think this is visible to all of you. And first of all, thank you so much for inviting me to your webinar today. I will try to speak slowly in English. I know there are many complicated and new English words. I wish I spoke Spanish, but thank you so much to our interpreter. And my task today is to provide a bit of background around what's happening with chlorpyrifos under the Stockholm Convention. I will not go into the health effects so much or any more details. I think Megan will do that in the next presentation. But I do want to talk a little bit first generally about the Stockholm Convention. Many of you I know is well familiar with this convention. It is the one internationally legally binding agreement that we have to control some of the worst chemicals that are around, the persistent organic pollutants. So these are the toxic chemicals that almost never break down through natural means in nature, and also can build up in your bodies and in the bodies of different mammals. And the Stockholm Convention addresses these persistent organic pollutants that are also transported long ranges to remote locations. The Arctic is especially mentioned as one of the especially impacted regions from these POPs. And the way that the Stockholm Convention addresses these chemicals is by adding them in an annex to the convention. So it's usually called that they are listed under the convention. So if they are in Annex A, the first annex, that means that the governments who are parties to this convention have decided that these chemicals need to be eliminated globally. There can be exemptions for different specific uses, but the overall goal is to get rid of them globally. There is also an annex specifically for persistent organic pollutants that are not intentionally produced, so unintentionally produced POPs. These are the ones like dioxins that forms maybe when you burn things or you produce other things. There are, I think, 7 listed right now. There is also a weaker annex where DDT and PFOS are right now listed, and that's what's called the Annex B for restriction. It means that it's not as ambitious as Annex A. It means that we want to get rid of it eventually, but at a slower pace. And looking at the convention today, there are 186 governments or countries that have signed on. And as I'm sure you already know, the US is not one of them, so they are not a party to the convention. We have 30 entries. You could call them chemicals listed under the convention, but I do want to emphasize that of these 30 chemicals or entries, some of them are actually big groups. So it's not only 30 specific chemicals, it's bigger than that. And there is a conference of the parties coming up in May 2023. And the 2 chemicals that will be discussed there will be another pesticide. It's methoxychlor, and also a plastic additive. And both of them have been recommended for listing in Annex A, so for global elimination. And there are 3 other substances or groups of substances that are under evaluation under the convention. It's a flame retardant called dichlorane plus. It's a group of chemicals that's called the medium chain chlorinated paraffins. There are also flame retardants, also used in plastics. And then, of course, our topic for today, chlorpyrifos. The POPs Review Committee, it's usually called the POPROC for short. It is the scientific body of the convention. So that means that it is the group of people who evaluates these chemicals and see if they fulfill the criteria for being listed under the convention. And there are 31 government designated experts representing all regions. They are not supposed to represent the political opinions of a country. But of course, as you all know, politics always come into play at these meetings. So you will, of course, have people at the POPROC who are scientists, but still have to carry certain opinions. There are also observers. So civil society, like we are, will be observers. That means that we can talk at these meetings, but we are not the decision makers. And that is true also for the countries and governments that are not actually part of the committee itself, but also of industry. So usually at this meeting, you have these 31 government representatives, but then you have more than 100 observers taking part in the conversation. And when the POPROC or the POPs Review Committee makes this evaluation, it happens in 3 stages. So it usually takes 3 meetings once every year to move forward in this process. And here is a photo of the most recent meeting of the POPs Review Committee. So this is what it looks like right now. The review process, it starts with that a country nominates a chemical for being listed under the convention. And that nomination needs to include certain amount of information. And based on that nomination and that information, there is a start of evaluation by the POPROC. And it goes through 3 stages, will I come back to. And then at the end of this evaluation, it's up to the POPROC to recommend a decision to be taken at the conference of the parties. So I know this is very technical, but I think it will be important just to help paint the picture of where Chlorpyrifos is right now. So bear with me a little bit longer, and we'll get into the substance itself. The first thing that happens with a nomination is evaluation if it actually fits the criteria under convention. So is it persistent? Is it bioaccumulating? Is it toxic? And does it transport to far distances to remote locations? If the answer to all of those decided by the POPs review committee is yes, then the chemical moves forward to the next stage. And the next stage is going from this initial nomination to a bigger evaluation that's called a risk profile. And in this risk profile, there's most of all, it's providing more information about all these criteria. And the whole idea of the risk profile is to provide as much evidence as possible to show that there is such a risk so that global action is needed. And that includes sources of release, it includes hazard assessment, it includes monitoring data, exposure data, and so on and so forth. And I think it's important to underscore here that this is not a typical regulatory risk assessment, where you compare concentrations found in nature with a certain safe threshold or anything like that. This is a convention risk profile. So it's more based on hazard. Even though you have monitoring data, it's not that you have to fulfill a certain number, if the concentration found in polar bears is higher than this or something like that. It is to provide a big picture of the risk of this. It's not made to quantify anything. And if the POPs review committee agrees that there is such a risk so that global action is needed, then this chemical moves forward to the next stage of the evaluation. And that is how to manage this risk. It's called a risk management evaluation. And so the final stage is evaluation of the risk management evaluation that has been prepared. So that evaluation includes much more information about how do you control this? Do you have to have an international control, like listing under the convention, or is national control sufficient? What's the different options? Are they effective? What's the benefits of this? What's the alternatives? What's the waste implications look like? Will you have big stockpiles around in countries that needs to be destroyed, and so on? It also has very important provisions around access to information about this chemical and public education. And since this is the final stage, this is also where the POPs review committee is expected to recommend a listing in Annex A, if we're lucky, in Annex B, if it's not the best choice, let's put it that way. And if it's unintentionally produced, then there could be a listing in Annex C as well. But the strongest listing recommendation is really listing in Annex A without any exemptions. Chlorpyrifos was nominated by the EU, actually in 2022. This has been a bit weird year when it comes to the POPROC meetings, because we've had 2 meetings this year to catch up there was no meeting in 2020 because of the pandemic. So they had to have 2 meetings this year to get back on track. And as you all know, this is widely used. It's an organophosphate pesticide. It's used in agriculture, but it's also used as a biocide, used indoors and outdoors in many countries. The first step of evaluation started in January this year. So that meant that the POPs review committee assessed if this chemical, this pesticide, fulfilled all these screening criteria. And I've copy-pasted a slide from the nomination, actually, where the EU showed as a summary that it is persistent, it does bioaccumulate, it's transported it has long-range transport, so that's the LRTP, long-range transport, and it has adverse effects. It's extremely toxic. And so there was agreement in the POPROC that the screening criteria were met. And it moved forward to the next stage. And that was the risk profiler. So that was evaluated in September this year. And this is where the complications escalated. They started already with some big fights in January. But in September 2022, at the POPROC meeting, this was discussed throughout the week and many sessions where most governments and most POPROC committee members agreed that there is such a risk, that global action is warranted. But there were a few countries that opposed this and said, first of all, they tried to say that we never agreed that the screening criteria was met, which is, you can't move back in the process. If the committee has agreed at one point, then the substance moves forward. You can't go back and start evaluating the screening criteria again. The review committee already agreed on this. So that was the first line of attack, so to speak. The chair of the committee was very clear on this, that this is already agreed, but this was a discussion that came up over and over again. But once we were able to move past that, there was another more difficult attack, and that is people started arguing that the levels that were found in remote locations were not high enough, so that there was such a risk that global action is warranted. So that would move us more into a risk assessment frame, which is not a convention frame, as we talked about before. It's really not the way the convention has worked before. It is clearly a way of attacking both the way the convention deals with things, but also it's very specific about chlorpyrifos, because there are some countries, specifically India was leading the attack on this. There were also other countries that were just trying to make sure that this does not move forward in the process. So the outcome of the meeting was that the committee could not agree that there was such a risk, so that global action is needed. But that doesn't mean that it's out of the process. It means that everyone is invited to provide more information to verify that there is actually a need for global action. So what happens now is there is a call for information where everyone, both parties to the convention, but also observers like civil society, can provide information to show why there is a need for global action. And the specific call for monitoring data and other type of data from remote locations, like the Arctic, but also if it's found in remote Pacific Islands, for example, that's also a remote location. And that kind of data is asked for. And that is to be gathered until the next meeting, which is in October or September, I think, next year at the 19th meeting of the review committee. And that was really all I had to say. I know I've been talking a lot of technical terms, so I also wanted to make sure that we had time for questions and comments around this. So I will stop sharing my screen and let's see if we have questions. Thank you.
Gracias, Sara. Las preguntas las haremos después de la siguiente presentación, pero podrían empezar a plantear sus
Thank you,
preguntas.
Sara. The questions we'll have after the next presentation, but people can start pasting their questions on the chat or the question and answer section. So, as we have seen, This is a discussion where all the countries have to take a stance regarding this nomination of Chlorpyrifos within the framework of the Stockholm Convention for global elimination. That's why we're organizing the second seminar regarding the problems of Chlorpyrifos on environmental health, presenting evidence for its facing this in different countries. We also have today an expert, Dr. Megan Horton. She was one of the experts who was able to make a presentation at this committee of experts. It's my pleasure that she's here with us today, Megan, and we will read some part of her curricula. I can't activate my video. Can you help me, please? Well, let's continue anyway. Introducing Megan Horton with some information. She is an environmental epidemiologist with experience of the impact of chemicals that are neurotoxic during pregnancy and early childhood regarding the development of the brain and behavior. Her lab in Mount Sinai in New York integrates exposure science, environmental epidemiology, neuroimaging, and advanced statistical approaches to understand how our environment affects human health. During the last decade, she has focused on the use of MIRs to study changes in the brain functionally and structurally in association to exposure to neurotoxics, including the application of pesticides that include Chlorpyrifos and their effect on neurodevelopment on children. She's currently an associate professor in the Department of Environmental Medicine and Public Health in Mount Sinai, and her lab is funded by the National Institutes of Health and the National Institute of Occupational Health. So we want to welcome Dr. Megan Horton, and we ask her to take the floor. Megan, can you activate your camera so we can see you and your presentation? Can you share your presentation?
Hi, can you confirm that you can see my presentation screen?
Sí, la vemos muy bien. Gracias, Megan.
Thank you. And thank you to everyone for the invitation.
Yes, we can see it.
I'm honored to present, and I think my talk today dovetails very well with Sara to combine the neurotoxic developmental effects of chlorpyrifos with what she discussed regarding policies at the Stockholm Convention to try to restrict its use. So just a quick reminder, I'm an environmental epidemiologist. I'm at IPEN School of Medicine in Mount Sinai, and my expertise is really understanding how early life exposure to neurodevelopmental toxicants, such as chlorpyrifos, can adversely impact the developing brain, resulting in persistent adverse outcomes throughout the life course. I'll start my talk today with a discussion of chlorpyrifos. I'm sure many of you are familiar with this compound, and as Sara gave a definition of the compound, I'll just briefly review why we're interested and concerned about continued use of this compound. Chlorpyrifos is an organophosphate insecticide that was originally developed as a nerve gas agent and used widely in warfare. In 1965, it was registered as an insecticide in the United States to replace DDT, which was a very effective but also very toxic pesticide. This was considered a regrettable substitution. This registration in 1965 was well before TSCA, the Toxic Substances and Control Act of 1976 in the United States, which provided the Environmental Protection Agency with the authority to require reporting, record keeping, and testing requirements and restrictions related to chemical substances and/or mixtures of chemical substances. So this registration was prior to those regulations. Rapidly, chlorpyrifos was used in the United States for agricultural, non-agricultural, and residential uses. In agricultural, it was used on corn, soybeans, cotton, citrus, almonds, among many other fruits and vegetables. In non-agricultural uses, it was widely used in golf courses, hospitals, roadway passages and right-of-ways. And in residential settings, it was widely used in houses and in apartments to control cockroaches and mosquitoes. It rapidly became the most widely used residential and agricultural pesticide in the United States until regulations by the US EPA in 2001 and then recent regulations last year that restricted regulatory use. In 2000-2001, chlorpyrifos was found in 91% of US homes in a nationally representative sample of the US population. Despite the success of the US EPA regulatory restrictions reducing this use, it's still widely used worldwide. In this recent publication, Hertz-Picciotto et al. Leveraged the most comprehensive and timely global database on recent pesticide use, and it include information reported by 71 countries in 5 regions. So in this map, you'll see the 5 different regions where these 71 countries are located. And the legend on the left demonstrates the average annual tons of organophosphate pesticides, including chlorpyrifos, used throughout 2010 and 2015. The darker the red color, the dark maroon color, represents regions where there are over 3,000 tons of chlorpyrifos applied annually in these regions. This demonstrates widespread use of organophosphate pesticides and includes use by agriculture, as well as use in homes, parks, schools, hospitals, and golf courses. This widespread use has clearly resulted in ubiquitous human exposure worldwide. While occupational application of chlorpyrifos is one source of exposure, particularly among occupational workers who are not using appropriate PPE, concern for neurodevelopmental toxicity stems largely from non-intentional environmental exposures, especially when we're looking at exposures among children. Babies and developing fetuses are exposed to about 5 times more chlorpyrifos than what the US EPA standards deemed safe, and children consume chlorpyrifos at 11 to 15 times the EPA standards. People can be exposed to pesticides by eating them, breathing them in, or getting them in their skin or their eyes. So you could be exposed if you're using pesticides in your own home. If your parents work in a setting or your household members work in a setting where pesticides are used, if you live close to a farm, or if you live close to a region that is using pesticides that have gotten into the drinking water. And then if you're not eating organic vegetables, which have been shown to have reduced levels of chlorpyrifos, there's considerable ingestion of chlorpyrifos through non-intentional ingestion. One of the concerns about chlorpyrifos, particularly aerosolized chlorpyrifos, is that chlorpyrifos is both very volatile and very stable. And so when it's used in a house, if a mom or a child or a family member sprays chlorpyrifos in the home to control for cockroaches, that pesticide stays in the air because it's designed to distribute within that household setting and stay in the air before it settles onto surfaces in the home and sequesters into the dust. This increased aerosolization of this pesticide increases the potential for exposure and we're talking about young children who are crawling around on the floor and often have increased hand-to-mouth activity. We're significantly increasing their risk for exposure. This is important because children are not just little adults. Their physiological and behavioral characteristics render them more vulnerable to environmental insults to compounds like chlorpyrifos. There's increased vulnerability in these developing fetuses and young children, largely because chlorpyrifos is known to pass both the placental and blood-brain barriers. We've demonstrated evidence of this in the Columbia Mothers and Newborn study, which I'll talk about a little bit later, when we measured levels of chlorpyrifos in mom's blood and in umbilical cord blood samples collected at delivery. What we saw was that umbilical cord blood samples often had higher levels of chlorpyrifos than the moms from which the babies were born. This is demonstrating that if the mom is exposed to chlorpyrifos, that chlorpyrifos can cross the placental barrier. As the fetus has underdeveloped detoxification and repair systems, she's unable to metabolize that chlorpyrifos, increasing her exposure and her risk for adverse outcomes of effects. The other thing that renders young children and the developing fetus more vulnerable is this idea of critical windows of development. An organ that is undergoing rapid development and growth is more vulnerable to perturbation. And if we think about the developing brain, these first few years of life, from gestation and the prenatal period and these first few years of life, there's rapid and dynamic development of the brain that sets the framework for how this brain will work later on. So this is the idea of critical windows of vulnerability, or critical windows of development that are more vulnerable to perturbation by neurodevelopmental toxicants. Another thing to consider with children is that there's a longer time for adverse effects to develop. So if you expose a child early in life, it has decades and decades for which an adverse health effect could occur. This is different than if we're thinking about an adult who's already lived many of their decades of life, and also their brain development is much more stable. An additional fact about children is that they have increased consumption per body weight of many contaminated foods, such as apple juice, fruits, and dairy. And they have a higher respiratory weight per body weight. So they breathe more, getting more potential exposure into their lungs. And as I mentioned, they have different behaviors. So increased hand-to-mouth activity, they're crawling on the ground, so they're lower to exposure regions once the chlorpyrifos has sequestered out of the air into the ground. Once we have an exposure in a child or developing fetus or in a mom, what is happening that might be affecting the brain development in the children? What are the mechanisms of action of chlorpyrifos? The most widely accepted mechanism of chlorpyrifos systemic toxicity, and largely its insecticidal properties, are due to the irreversible inhibition of acetylcholinesterase. So a quick schematic of acetylcholine and acetylcholinesterase to help us understand the implications of this mechanism. Acetylcholine in this green panel is a neurotransmitter that communicates signals between neurons in the central nervous system and the peripheral nervous system. Acetylcholine has numerous functions in the body. It can be found in all motor neurons, where it stimulates muscles to contract. It controls movements from the stomach and the heart to the blink of an eye. All of the body's movements involve actions of this important neurotransmitter. Acetylcholinesterase is the enzyme that breaks down acetylcholine to terminate this neurotransmission. So ideally, in a functioning system, once the impulse has been propagated across the synapse, so the activity has moved, so the impulse has moved from the presynaptic neuron to the postsynaptic neuron, acetylcholinesterase is designed to break down that acetylcholine, terminating the transition. So acetylcholinesterase breaks down or prevents the action of acetylcholine. And in acetylcholinesterase inhibitors, such as chlorpyrifos, prevents this from happening. So acetylcholinesterase prevents an acetylcholinesterase inhibitor, such as chlorpyrifos, prevents the breakdown of acetylcholine, prolonging that stimulation or contraction. So this is the action, this is the mechanism that's associated with insecticidal activity. So if you spray a pesticide and an insect is exposed, the insect has this prolonged stimulation or contraction, which results in an immediate paralyzation and ultimate death of the insect. The effects of this are different in the developing brain. However, the mechanism of acetylcholinesterase inhibition is persistent, is consistent across cockroaches and other insects and humans. It's the same biological mechanism. One important thing to note is that it's fairly high levels of chlorpyrifos that have been associated with acetylcholinesterase inhibition. There is a threshold effect. There's a level at which higher than this threshold level, acetylcholinesterase effects are seen, and there's a level that is considered lower than the dose to stimulate acetylcholinesterase inhibition. When we're thinking about neurodevelopmental toxicity of chlorpyrifos, it's been associated with lower doses of chlorpyrifos. doses that are too low to stimulate the acetylcholinesterase effect. The US EPA in 2016 stated that there was sufficient evidence that there are neurodevelopmental effects occurring at chlorpyrifos exposure levels below those that are required to cause acetylcholinesterase inhibition. This is very important when we're considering legislation and regulation for chlorpyrifos, that we look at levels and mechanisms of action that are occurring below those levels that stimulate acetylcholinesterase inhibition. So several of the mechanisms that have been associated with lower dose exposure to chlorpyrifos include disruption of DNA and RNA protein synthesis, disruption of other receptors other than acetylcholinesterase, changes in enzyme activity, changes in signal transduction, neural differentiation, and the generation of reactive oxygen species and oxidative stress. Very critical to the evidence provided at the Stockholm Convention is the suggestion that low-dose chlorpyrifos acts to disrupt thyroid hormone action, or it acts as an endocrine disruptor. A quick reminder of the importance of the hypothalamus pituitary thyroid axis, or the HPT axis, is here in this figure, looking at the role of thyroid hormone and brain development. So the HPT axis primarily functions to maintain normal circulating levels of thyroid hormone. Thyroid hormone is essential for the biological function of all tissues, including regulation of many organ systems and functions and energy expenditure. For example, it regulates cardiovascular function, bone function, liver function, can influence food intake and energy expenditure, among many others. Another primary function of the HPT axis and thyroid hormone is regulation of brain development. So here you have the pathway of the HPT axis, so the hypothalamus to the pituitary to the thyroid, and how that could ultimately impact brain development. Thyroid hormones are essential for the development of the mammalian brain in particular, acting on migration and differentiation of neural cells, synaptogenesis, and myelination, as demonstrated in this figure. All of these activities are very prolific and very active during this period of gestation, all the way through the early childhood period, and somewhat up through adolescence. Disruption of these processes at multiple levels along the HPT axis during these critical windows of development, particularly during early pregnancy, can lead to deleterious effects on the offspring. So there is some evidence in animal models suggesting that chlorpyrifos exposure at dose levels not inducing acetylcholinesterase inhibition causes thyroid hormone alterations in mice, dams, and offspring. Further, gestational exposure in mice has been associated with significant reductions in brain thyroxine levels from juvenile stages till adulthood, hinting at the persistence of these effects. And finally, short-term low-dose oral exposure in female rats increased thyroid hormone levels, and changes in thyroid hormone levels in moms can affect the thyroid hormone levels in the offspring. In humans, what kind of developmental neurotoxicity are we seeing? In the late 1990s, an investigator named Gillette and colleagues overcame many of the difficulties of studying pesticide exposure by enrolling children in 2 communities living in northern Mexico. These 2 communities were similar in all aspects, except one community was highly exposed to agricultural pesticides, while the other was not. As part of this assessment, children who were 4 to 5 years of age, were asked to draw a person. You can see a demonstration, an example of a figure on the left, which was drawn by a child who was from one of the non-exposed communities, compared to the figure on the right, which is a child from a similar age, same-sex child that was drawn by a child who was exposed to chlorpyrifos prenatally. While this pioneering study was criticized for its epidemiologic design, reviewers suggested that they were not adequately matched, that there were other things that may be causing this effect. Its compelling results led to the recent decades of well-designed epidemiologic and experimental studies demonstrating the adverse effects of early life chlorpyrifos exposure on children's health. In addition, there have been several systematic reviews and multiple epidemiologic studies that I'll talk about briefly. These reviews have shown These reviews notably are from the US and from other countries spanning diverse populations in both urban and agricultural settings. And they all link organophosphate exposures, in particular chlorpyrifos, during the fetal and childhood development with poorer abnormal reflexes in newborns, with mental and motor delays among preschoolers, with decreases in working and visual memory, processing speed, verbal comprehension, perceptual reasoning, and full-scale IQ among elementary school children. And also studies that have linked early life chlorpyrifos exposure with elevated risks for symptoms or diagnoses of developmental disorders such as ADHD and autism spectrum disorder. Notably, levels of exposure in these studies were too low to induce measurable depression of acetylcholinesterase. So these are happening due to those other potential mechanisms of action, such as thyroid disruption. And as I said, these are happening in diverse populations in both urban and agricultural settings, and they're happening worldwide. In one review, the adverse effects of organophosphate pesticide exposure were seen in all but one of 27 independent studies evaluated, with the strongest associations occurring following prenatal exposure. This leads me to one of the critical pieces of epidemiologic evidence demonstrating the neurotoxicity of prenatal chlorpyrifos exposure that we performed in children living in New York City.
Megan, can you speak slowly, please?
Sure, thank you. In 1996, New York State was one of the first states in the United States to pass a pesticide reporting law. For the first time, this allowed for the first time in New York State and in the United States, we were able to generate a baseline description of the type, relative intensity, and geographic variation of pesticide use. Using this data, so this map is showing the map of New York State, and the red dots represent the gallons of pesticides used by country in 1997. So the larger red dots are the highest amount of pesticides applied by gallons or pounds in these regions. This is data from a 1998 study of pesticide use in New York State that found the heaviest application in gallons and pounds of legally registered pesticides by licensed applicators occurred not only in agricultural regions, but in the boroughs of Manhattan and Brooklyn in New York City. Chlorpyrifos was the insecticide most heavily applied in New York City and one of the insecticides most widely used by pest control operators in the New York City Housing Authority. So what this map shows and what this new data showed was that the highest use of pesticides in New York State was not necessarily in the agricultural regions, but in the highly urbanized populations in New York City. In fact, there was more chlorpyrifos applied in New York City than in all other New York counties. This information about widespread exposure, both in agricultural communities and in residential communities, was released right about the time that the Columbia University Center for Children's Environmental Health, one of several NIH-funded centers focused on children's environmental health, was initiating the longitudinal birth cohort study, the Mothers and Newborn Study. The Mothers and Newborn Study enrolled over 700 African-American and Dominican women and newborns living in northern Manhattan and Washington Heights in the northern regions of Manhattan. So this is a map of Manhattan, of New York City. This long rectangle is Manhattan, and up here is the North Bronx. And the blue dots represent where the moms were enrolled, where they were living. Notably, if you look at the colors, this is also the map of where the moms were enrolled from overlap with a map of poverty in the in New York. So the darker colors, the darker gray, represents a higher percentage of the population that's living below federal poverty lines by quantiles. So we enrolled women from low socioeconomic settings that were largely living in public housing or housing with disrepair. We collected environmental and biological samples during pregnancy from the mother and followed the child throughout childhood and now throughout adolescence. The main goals of the study were to evaluate the effects of environmental exposures, including pesticides, on children's neurodevelopmental outcomes. Upon enrollment into the study, it was revealed to us that 90% of the mothers, so these are pregnant moms, they reported using pesticides during their pregnancy. Many of them were using the spray cans containing pesticides such as chlorpyrifos during their pregnancy. One of the fascinating and useful things about the mothers and newborn study is the timing of enrollment that was coincident with some of the US EPA regulatory action restricting residential chlorpyrifos use. So in 2000-2001, based on animal data and laboratory data, the US EPA took regulatory action to phase out the residential use of chlorpyrifos. So it was no longer allowed to be used in the spray cans, bombs, etc., that were used within homes to control cockroaches. As I mentioned, the moms in the Columbia Mothers and Newborn Study were enrolled between 1998 and 2006. The US EPA regulations came in after about a third of those moms were enrolled into the study. So this set up a sort of natural experiment where we could look at whether or not moms who were using pesticides prior to the regulations had higher levels of chlorpyrifos compared to the moms who were using pesticides following those regulations. What we showed is that EPA regulations were quite effective at reducing exposure to chlorpyrifos in this population. In this small graphic on the left, we're looking at levels of chlorpyrifos measured in personal air samplers that the moms wore during pregnancy. So during this, it was a 3-day period in their 3rd trimester of pregnancy. Moms wore a small backpack with an air filter at their breathing zone. The air filter collected air that was similar to what the moms were breathing, and we used that to measure chlorpyrifos in their breathing zone. You can see the levels of chlorpyrifos in the blue bars by year of enrollment, 1999 all the way up through 2005, with the red arrow indicating when the US EPA regulations took place. One of the things that this clearly demonstrates is that there was considerably higher exposure to chlorpyrifos in personal air samples prior to the regulations, and it went down significantly following those regulations. On the right, we're looking at chlorpyrifos levels measured in umbilical cord blood samples that were collected at delivery. Again, looking at 1999 up through 2004, looking at the picograms per gram of chlorpyrifos in cord blood over time. In the moms that were enrolled in 1999, there were considerably higher levels of chlorpyrifos detected in cord blood. And by the time we got to 2004, after those regulations, chlorpyrifos levels in umbilical cord blood were non-detectable. So this is evidence of the efficacy of residential restriction of chlorpyrifos use on exposure. So what about on the health effects associated with early chlorpyrifos exposure? One of the things that we've done at the Columbia Newborn Study is look at the effects of early life chlorpyrifos exposure on intelligence using the Wechsler scales of intelligence for children at 7 years of age. So this slide is showing the association between chlorpyrifos exposure and IQ in 7-year-old children. On the x-axis of all of these figures is chlorpyrifos levels in picograms per gram. And on the y-axis is the results of the association between chlorpyrifos and performance on IQ measures, including full-scale IQ, working memory, verbal comprehension, perceptual reasoning, and processing speed. What we're looking at in these smooth cubic splines are the slope between that association. So you can see with increasing chlorpyrifos exposure, there was decreasing performance or lower IQ in these 7-year-old kids. This effect was most profound in the working memory association. To translate, that means that for each standard deviation increase in chlorpyrifos exposure, full-scale IQ declined by 1.4% and working memory declined by 2.8%. One might think that these are not strong or large effects, and we admit that these are small effects. And if you were to look at these effects in an individual person, you might not see an association. It's not as though you could identify a children or that this was associated with a diagnosis. But what's important in these epidemiologic studies is to think about the impact of this 2 to 3 point decrease in IQ on a population level, and the impact of shifting the curve slightly towards the lower norm. So this could have a profound effect of shifting more kids into the area of kids who are needing special help or have learning disabilities. And it shifts the other side of the curve down, where there are fewer kids optimally performing at grade level. And these are not dissimilar. These are similar to the effects that we've seen associated with low-level lead exposure, and also with things like environmental tobacco smoke exposure. So while these effects are small, they're still very significant, and on a population level could have a considerable burden. Very importantly, these results were observed in other longitudinal studies of children's health, demonstrating the generalizability and the robustness of these associations. In the Chamaco study in California, which is the longest running study of pesticides and environmental exposures in a farm worker community, investigators saw similar results between chlorpyrifos exposure, which again is on the x-axis, and performance on performance on IQ outcomes in the same age kids. So again, you'll look at this panel in panel A, a decrease in working memory performance with increased pesticide exposure, which is largely led by chlorpyrifos. There's a third study evidencing the effect of chlorpyrifos on outcomes, IQ outcomes. So this is looking earlier in life. This is looking at the Bailey scales of infant development at 12 months of age. And you can see we're focusing right here on full scale IQ on the left, perceptual reasoning on the right, both different domains of IQ. And if you look at these black triangles, you can see that it's the highest tertile of chlorpyrifos exposure, or metabolites of chlorpyrifos, that are associated with the greatest decrements in full scale IQ and the greatest decrements in perceptual reasoning. So these are 3 epidemiologic studies in different populations using different methods that all came up with the same results, that early life, particularly prenatal chlorpyrifos exposure, had persistent effects on IQ in these children. Adding to the behavioral data, we provided critical support for the association between early life chlorpyrifos exposure and brain development by using MRI, or magnetic resonance imaging. Magnetic resonance imaging allows us to look inside the brain to look at structural and functional changes that may be associated with early life environmental exposure. To do this, we enrolled a subset of children from the Columbia Mothers and Newborn Study. We picked 20 kids who were most highly exposed to chlorpyrifos, so all of whom were in the highest quartile of exposure. And we picked 20 kids who had no chlorpyrifos exposure and additionally had very little environmental tobacco smoke exposure and no air pollution exposure, or little air pollution exposure. And we compared the brains after matching for sex and age. What we see here in this is a sagittal view of the brain. So we're looking at the brain from the side, looking at the right side and the left side of the brain. And we saw that prenatal chlorpyrifos reduced the prefrontal cortex volume in areas of the brain that are most widely linked to higher cognitive functions, such as working memory. So in the prefrontal cortex, the orbital frontal cortex, and the retrosplenial cortex, all regions of the brain that may be the home for higher executive functions, such as working memory. Also, and though this is a small sample size, we do see some suggestion of a dose response that chlorpyrifos has on surface measures in the brain. So this is looking just within those kids that are most highly exposed to chlorpyrifos, and seeing that the higher you're exposed, the more different your brain is than a control brain. So in conclusion, based on this epidemiologic data combined with the prior laboratory data, we demonstrate that low levels of chlorpyrifos are damaging to the developing brain, that the adverse impacts include delayed motor, cognitive, and behavioral development and brain structural abnormalities, and that these effects may be persistent or permanent over time. Based on this sound science, along with 2 decades of risk assessment and input from science advisory panels, in 2001, the US EPA scientists concluded that chlorpyrifos is unsafe at any detectable level and banned agricultural use, adding to the existing residential bans. So if I have just a few quick minutes, I can tell the story of the regulatory action in the United States and talk a little bit about what we might do so that it doesn't take quite so long for other chemicals. But just briefly, it was in 2000, under the Clinton administration, that the US EPA first announced an agreement with Dow, one of the primary producers of chlorpyrifos, to end home and garden use, leading to those 2000-2001 restrictions. In 2011-2012, the data that I previously presented was generated in human epidemiologic studies, bolstering the science that we had from animals. In 2015, under the Obama administration, the EPA proposed a ban on food uses of chlorpyrifos, so agricultural uses, because they found it did not meet the safety standards. We were optimistic for a federal ban on agricultural use at this point, but at 2017, under the Trump administration, the EPA refused to finalize this ban, despite the inability to meet federal safety standards. Between 2018 and 2021, under the leadership of Earth Justice and many environmental advocacy groups, we went state by state to try to get regulations, and were successful in Hawaii, Maryland, California, Oregon, and New York, and close to successful in several other states before 2021, when under the Biden administration, the EPA finally finalized the proposed agricultural ban that has led now to the success in the United States that we no longer allow any detectable levels of chlorpyrifos in agricultural products or residential use. I was asked to comment somewhat about why this took so long, and what are the flaws in risk assessment that don't account for children's vulnerabilities. I think there are several things to discuss, and we can maybe have more time in the questions. But one is that the US EPA is only now considering to revise the way it uses scientific research in policymaking. Under the proposed policy, scientific research that requires personal health information and raw data cannot be used for lawmaking. This applies to a lot of pesticide research and other exposure research, and it keeps information like the Columbia and Berkeley studies out of risk-making decisions. The role of science at EPA will be a topic of continuous discussion and will have broader implications on how we safeguard the environment and human health. The other thing to think about is that many risk assessment outcomes are based on diagnoses or clinical findings, but much of what we see in children are subclinical findings or low-level effects, which, while may not have an effect on an individual, would certainly have an effect on the population. Another consideration or particular flaw in risk assessment is that that many chemicals act non-monotonically or not linearly, particularly when we're talking about endocrine disruptors. So they act on a U-shaped curve where a very low level effect may be toxic and a high level exposure may be toxic, but exposures in the middle range are not seen to be toxic. And how is this accounted for in risk assessment? For children, it's important to consider the time lag between exposure and outcome. As I mentioned previously, these are very young children with a long time to live, and we very rarely see an immediate impact of low-level exposure. Rather, we see these persistent effects on neurodevelopment when we look at trajectories over time. Final considerations could consider the long-term chronic exposure throughout early childhood and then additional critical windows in adolescence. We rarely account for the mixture of toxicants, but we know that kids are not exposed to chlorpyrifos alone. And this multi-hit or mixtures exposure could have many different effects than the individual chemical alone. And we need to account for non-chemical exposures, such as the social environment, that we know impact or can exacerbate the effects of environmental exposures on neurodevelopment. So with that, I'd like to give a brief thanks to the many researchers and the families who participated in these studies, particularly at Columbia University, at Icahn School of Medicine at Mount Sinai, and at the University of California, Berkeley, funding from the National Institutes of Health, and Earth Justice, which is an advocacy firm which helps us with the statewide and national regulations.
Muchísimas gracias, Megan, por esta excelente presentación, y ahora les pido, por favor, si pueden.
Thank you deeply, Megan, for this excellent presentation.
Pueden activar su cámara.
So, can you activate your camera?
Not yet.
Todavía no puedo.
Megan tiene problemas, si le pueden ayudar.
Megan's having problems. Can you help her with the camera?
A ver, Megan, can you try?
It still says
Let's see,
the whole.
Megan. It's
A ver.
Ah.
Perfecto.
perfect. There you
Got it.
are. Very good. Now we can see you.
Great.
Well, these two presentations have been very interesting. Between the risk assessment, the conventional risk assessment, and the risk profile based on the hazard that is the requirement of, that is part of the review process of the pop rock, Sarah.
Thank you very much. That's an excellent question and a difficult topic, but I will try to give a brief overview. I'm also going to post a link to a Spanish translation of a risk assessment guideline if anyone is interested to learn more details. I found one that is developed by the Swedish Chemicals Agency. So that's the government agency that deals with chemicals here in Sweden, which is the guidance is pretty good. It explains well. But in general, risk assessment is usually done from regulators to assess whether a chemical can be used in a product or used as a pesticide or in cosmetic or anything like that. And the first step is the same as in the POPs review committee. You look at the toxicity, the hazard of that chemical. Is it toxic? When it comes to the regulatory aspect, you typically look at some, if it's for environmental damage, for example, or hazard, you usually look at different type of species. So you can look at some water living organisms like fish and some soil living organisms like different worms and so on, several different types of organisms. And you see how much of this chemical is needed to bring on a specific toxic effect. So if you expose this worm to a series of concentrations of these chemicals, when do you start seeing different types of effects? Usually it could be for small organisms like worms, it could be death as the thing you're looking at. And there are many different varieties of that. And then you take that concentration where you see effects, and then you say that everything below that concentration is safe. And as Megan just explained, that is not the case. That's one weakness, but that's how the regulatory system works. And then when you assess the uses of that chemical, you look at exposure. So then you compare how much if you're looking at something that will end up in soil, you look at the concentration in the soil compared to that level, that threshold that you consider safe. And roughly, if you say if it's above, then you have a higher risk. And if it's below that threshold, it's safe, more or less. So that's a regulatory perspective. You want a number. You want to say yes, no. When it comes to the POPs review committee, you're not asked for that number. You're not required to produce that type of number. You look at everything related to that chemical. So for chlorpyrifos, you would look at every monitoring data that you have from all types of species. You will look at levels in soil, in ice, in water, wherever you find it, in humans, of course. And then you will just, you will have a conversation around, is this acceptable? And of course, it's not when it's such a toxic chemical, but you don't need a specific number. So you make more of an overall judgment based on all the data that you have. So you don't base it on a specific number. When it comes to the Stockholm Convention, you base it on all the available evidence that you have, that it's both harmful and it exposes people in locations far away from where it was released. That is a very, very brief version.
Muy bien, Sara. Otra pregunta. Hay un
Very good,
comentario.
Sarah. Another question. There's a comment by Carmen Ciranda a colleague from Uruguay. It's a comment, and then there's a question. She says, vis-a-vis the critiques of the COP review committee, such as that the selection criteria were not complied with, or that the criteria, the data from faraway places, do you think this is anti-ethical or immoral? I think we could back this with a joint statement to the scientific committee signed by professionals and scientists. My question is, how do you choose the experts on the pop crop committee to avoid these experts, to ensure that they're objective and that they're not representing economic concerns, especially if they come from the countries that are the main producers of chlorpyrifos in the world, like China and India? That's the question, and comment.
Excellent comment and question. I think having some joint statement, I think that's a great idea. I would say that this is a political issue. It's not about the evidence. The evidence is there. But when people want to oppose something, they use whatever means they can. So that's a comment on the first comment. I would say when it comes to the members of the committee, there are certain procedures they have to go through. They have to show that they are not dependent or affiliated with any of the actual producers. So there is a conflict of interest procedure that they have to go through. And of course, it's also up to civil society and others that watch the committee to make sure that they don't have any ties to industry, as we can find. But then there is this also complicated issue that if you are coming from a country, you will have this pressure to have a certain opinion, and it is a weakness of the committee for sure. It's also very hard to get around if you don't have any evidence that one of the scientists are employed by or have been employed by or are in any way financed by the industry. It's very difficult to say that that person cannot be on the committee. But so I don't have an answer or a solution. I just acknowledge that yes, it is a problem, but there is certain procedures at least for conflict of interest.
Muchas gracias, Sara. Ahora tenemos, antes de tener las preguntas para Megan.
Before questions for Megan, we want to remind all the participants that on the chat, we have placed a link so that you can register to that link to receive the presentations of the presenters of this whole webinar. And so we can send you the recordings later on of this webinar, of this conversation that we're having today. So now, to Megan, a question. In many countries, chlorpyrifos, ethyl, ethyl, and methyl are registered. Chlorpyrifos methyl has been banned in many countries. I don't know whether chlorpyrifos methyl has many differences in relation to chlorpyrifos ethyl, whether they have there's any research that shows harmful effects on health or neurological development or other harmful effects. There's also a question for Sara, whether the nomination at the Stockholm Convention, whether there are radical differences in the nomination process. So another question would be whether you could elaborate more on this difference for the hormone disturbers when you say that there's not a linear dosage effect in the non-monotonic curve, whether you could explain this a little bit more in depth, what that means, because we cannot really refer to a safe dose of exposure. Can you delve more deeply into this topic, please, Megan?
Sure. I can't speak upon the difference of chlorpyrifos methyl versus ethyl because I understand that in the US, the regulations both on residential and agricultural refer to the chlorpyrifos parent compound. So I don't know the differences. And we didn't, in our biological and environmental measures, we didn't distinguish whether the toxic effects were due to the ethyl or the methyl species. Sarah might know more to the response to that. Regarding the question about the dose response, what we observed in a very small subset in the chlorpyrifos MRI study is a linear dose response between chlorpyrifos exposure and changes in brain volume in those prefrontal cortex regions. So this is suggesting that there was not a threshold effect, that while the effects were greater at higher levels, we saw effects at the whole linear dose response that were different than those without chlorpyrifos exposure. So with chlorpyrifos, between the animal data and the human data, it's clear that there is no threshold effect for the low dose neurotoxic mechanisms. What I mentioned in the last slide is that many chemicals, particularly other endocrine disrupting chemicals, do not necessarily follow a linear dose response where it's easy to identify whether there is a threshold or whether there is no threshold. In a non-linear or non-monotonic dose response, you see either a U-shaped curve like this or the inverted U-shaped curve, where you see health effects at either point, but you don't see effects at the medium levels. And this makes it very hard to establish whether there is a threshold because there's no cut point at which you see effects. There are multiple cut points at which you see effects. So I hope this answers the question about the relationship with dose response and a linear dose response and a non-monotonic dose response and endocrine disrupting chemicals in health.
Any opinion about the difference between the ethyl and methyl chlorpyrifos, Sarah?
Thank you. From a convention perspective, what was nominated was chlorpyrifos ethyl. So that's what we usually refer to as only chlorpyrifos. It did not include chlorpyrifos methyl, unfortunately, which means, and that has been discussed under the convention in the POPs review committee. There is less data on chlorpyrifos methyl, which is a problem, but also since this is a controversial chemical to discuss, the committee agreed in the end to only look at the chlorpyrifos ethyl. So that's what we call chlorpyrifos, and not include methyl in the nomination. I don't know in the end when it gets listed if that makes a difference. I think we will have to look more into that. But right now it's only chlorpyrifos ethyl that it's under discussion under the convention.
So we can still use Megan and Sarah's time. We will step into additional questions that are pending, and then I will make another presentation myself. So Megan, can you activate your camera, please?
Hi.
There's still a question. There's still a question pending. It says, how difficult was it to implement the regulations to ban chlorpyrifos by EPA? How difficult was it to implement this? And related perhaps, what role did the surveillance, the epidemiological surveillance play in this process? What role did epidemiological, did it play a role in the process to ban chlorpyrifos, and what role did it play if it did play a role?
As far as to speak to the excuse me, the implementation of the regulations, I don't know how difficult that was to do on an industrial level and residential level. I can just speak to the efficacy of that because we actually went around 2 stores in the Washington Heights area. We went to 250 different bodegas, grocery stores, hardware stores, any location that was known to sell residential pest control. And we pulled anything that they sold that was for residential pest control off the shelf. And out of those 250 stores, we only found one dusty can of chlorpyrifos. And this was just a year after the regulations. And so they were very rapidly pulled off the shelves for residential use and replaced with an EPA list of suggested replacements. Regarding how the epidemiologic surveillance was used, it tells to the last point that I discussed in the PowerPoint, that there is some contention about using NIH funded studies for regulation because of the concerns about privacy and protection of confidential data. So I think that the most influence was, I think there was an interesting balance between the environmental advocacy groups and Earth Justice leading the effort to change the market use. So as I think the strategy, particularly once in 2017, once the EPA ban that we had been optimistic for federal regulations was declined or denied by EPA, and we decided to go state by state, the strategy was to hit the market. So if we could influence states that were widely using chlorpyrifos in some of the biggest agricultural uses like California, that we were hoping that could influence Dow's decision to continue using chlorpyrifos. And it's anecdotal, but that did seem to work, because around 2019 is when Dow considered phasing out chlorpyrifos and stop producing chlorpyrifos in the United States and for use in the United States. So I think that all came from the epidemiologic studies that were not necessarily used directly in the EPA risk assessment, but that were used to shift the market for chlorpyrifos in the United States.
Thank you very much, Mabel. Yes, there's a legal struggle, very extensive legal. Jennifer Sachs participated in the earlier seminar of she referred to this legal battle where different groups of justice participated, and also part of the Pesticide Action Network in North America, and other groups participated. So scientific evidence is not enough if there's no political willingness, and political willingness, we have to use public awareness campaigns.